Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don and I'm a digital marketer. And before we get started today, I have just a quick message from our sponsor.
Being at the epicenter of the next breakthrough in biotechnology. That moment could happen for you this October at Bio on the Bayou 2025, where Southern innovation meets scientific excellence in the heart of New Orleans. Over 300 of the brightest minds in biotech are coming together for 2 extraordinary days in New Orleans. We're talking groundbreaking research from powerhouses like Tulane, Auburn, the University of Mississippi, Xavier University of Louisiana, and LSU Health. Expect revolutionary technologies, cutting-edge research, and industry-shaping expert panels that you won't find at any other event, plus networking opportunities that could transform your career. This isn't just another conference. It's an intensive 2-day summit where real partnerships form. We leverage the cutting-edge Partnering One platform to ensure that you'll connect with exactly the right people, whether you're seeking investors, research partners, or your next big opportunity. The future of biotechnology is being shaped right here in the broader Gulf South and having a global impact. Will you be part of it? Mark your calendar for October 28th and 29th. Head to bioonthebayou.com right now and secure your spot for the most exciting biotechnology event of 2025. The next breakthrough could be yours. Bio on the Bayou 2025. Register today.
In this episode of the Life Science Success Podcast, my guest is Michael Edwards. He's the founder and CEO of Bioinfo Solutions, a data and analytics company specializing in extracting insights from complex biological information. Welcome, Michael.
Well, thanks for having me. Good to be here.
Yeah, it's great. It's great to have you, and I look forward to our conversation. Before we get started, you mind telling everybody a little bit about your journey? So, what was it that brought you to the field of genomics and bioinformatics?
I've always been interested in science. I think it was grad school. I was on my 25th RT-PCR for the week, looking at one gene at a time. And what I basically went while I was doing this, microarrays came out, which is a way to look at all gene expression at the same time.
Mm.
So for me, I was like, why am I looking at one gene at a time when I can look at everything at the same time and get biological context? So, as soon as the microarrays came out, basically encouraged me to kind of pursue that area. I didn't really have a lot of statistics, computer background, so kind of had picked that stuff up. Actually, when I started, bioinformatics wasn't even a word. It was just, what do we do with all this data? So, I picked up some math, some computer skills, and kind of left the lab and been doing research on my computer ever since.
Yeah, I think in our initial conversation, the one thing that I think I told you before is that I kind of look at people that do bioinformatics as kind of the wizards of our time, because you guys sort of, you could walk in a room and the scientist in the room would say, look, we can't see what we're looking for. There's a lot of noise in here and whatever, and then all of a sudden, through bioinformatics and the changes that you can make in the data, you can filter out a lot of the noise and make it a heck of a lot clearer. And so I just, yeah, I mean, I'm always amazed, but at the same time, it's not that hard to understand that, hey, look, through proper filtering, we could see a lot more of the data too.
Yeah, totally. But sometimes you do your filtering and you don't get anything either, and that's the hardest conversation, right? Hey, I just spent $30,000 on this experiment, did all the sequencing, tell us what's going on, and you'll do it, and there's nothing there. So
Yeah. So tell me a little bit more about your journey. What have been some of the key milestones overall? I've seen, like, what is it, University of Wisconsin in your
yeah.
past. You're here in Colorado now, so just love to hear more about your overall journey and what the key milestones were.
Oh, sure. I started out, went to Colorado State University, got my degree there. Did a master's degree at the University of Texas Medical Branch in human genetics. Um, Worked on brain aging while I was in Texas. Took a year off. I think for a while I was working at a veterinary hospital as a lab tech. So that I'd have to go into the. We were actually my PI at the time was working on regenerating cartilage in racehorses.
Hmm.
so so I'd have to go in. They had this huge morgue at the veterinary school, and I'd have to collect cartilage from dead horses, which was a very interesting job. There was a lot of times there'd be like, cows, in the morgue. So it didn't smell very good, but did that for a while. Went on to get my doctorate degree at University of Wisconsin, and that was basically where I discovered the microarray technology. I think that was the thing that kind of changed the way I looked at everything. That it's I see a lot of labs. They focus on one gene. They write a lot of grants for that gene, and it makes you see everything from that lens, so that when you approach a problem, you're like, you know how does my gene interact with this, rather than look at the problem kind of fresh and say, what is the data telling me, that's going on. So I would say that was kind of a milestone. What else? I think the biggest thing for me was seeing how all this information collects. That
Hmm.
if you go into Gene Expression Omnibus, this is kind of a depository where we're putting a lot of our genomic, proteomic data. It just keeps getting bigger and bigger. And a lot of people kind of ignore this, and they're just doing new experiments. But if you work on breast cancer, there's already 1,000 experiments on breast cancer. Why not utilize that information? So I think, as I went in my journey and, saw that data accumulate and was able to use that to answer questions where I didn't have to actually get a grant or run an experiment, I think that was a game changer for me. So,
Yeah. And then in terms of your career, right? You've taught, you've done research, you've worked in industry. What motivated you to start Bioinfo Solutions?
the desire not to write any more grants. Oh, I hate writing grants. Yeah, in academia, I just, it's such a slow process, right? I have an idea. I write this grant. That takes months. Then you submit it, and it takes months to, get a response on that. And then it's months to get your money. And so for me, I just, got tired of that whole process and decided, I'd love to work on everything. And I think that was another thing too, is I've just been interested in so many different topics that it just didn't make sense for me to just focus in on one, I guess, if
Mhm.
that makes sense. So yeah, I think it was a frustration with the administration, just any kind of like rules, So, and there's plenty of rules in academia. So, went out
Yeah.
on my own and yeah, got to work on lots of different projects, which was really cool.
Yeah, and I mean, it's probably a really tough time, right now for companies that are primarily surviving, through working with academia, given some of the challenges right now with NIH and others. But I would imagine that most of your clients today are probably not in the academic world. So tell me a little bit about why somebody comes to Bioinfo Solutions, and how do they get started, in working with you?
Sure. Well, I do have a website, which not very many people go to. It's usually my YouTube channel. So um, I've been recording my lectures for about 10 years. I do have some fairly popular videos out there. If anybody works on the Cancer Genome Atlas, I've got a bunch of videos that are pretty popular on that. A lot of times they'll see my videos and reach out to me, and with a problem, or typically it's just word of mouth. A lot of times is, I work on a project, I do a good job for somebody, and in the biotech world, everybody moves from company to company. So, the same person I was talking to now, at one company is working at another, and hey, Mike, you did a great job for us when I was working there. Can you, look at our data here? So, and the projects I get can be everything. I've worked on cannabis. I've worked on medicinal mushrooms. I actually worked on for a client. It was mycotechnology, but there was there's a mushroom called the honey truffle, and it's a sweet mushroom. And somebody at our company or at mycotechnology decided, what makes this mushroom sweet? So, mushrooms typically don't produce a lot of sugar. So we went in, did a lot of research. Eventually, I found the gene for that sweet protein. So what we've ended up finding was it was actually a sweet protein, which means you don't have any diabetic risk, no calories.
Hmm.
So right now they're actually trying to commercialize that product. They're calling it honey truffle sweetener. So
Huh.
so be looking out there for a sugar substitute that's derived from mushrooms.
Wow.
I thought that was
Uh,
a really cool project because a lot of time I'm usually working on diseases and stuff, and there was like, hey, we know this thing makes this sweet. Here's a protein sequence. Can you now find the gene? So
so the honey truffle does not have psychotropic effects. Then I'm
no,
assuming.
and that's the thing. Everybody I say, hey, I worked for a mushroom company, and they're like, what state are you from? Colorado. I'm like, oh, those mushrooms. And I'm like,
Uh,
no.
right. Yeah. Well, I mean, it's funny because I've worked, I've done, some work for two different Canadian companies, and they were both researching, psychedelic mushrooms. So it was interesting to me as well that you have different people in different sort of areas of the world working on the same things, but for different reasons.
Yeah.
Oftentimes.
No,
So.
I think fungi are very interesting in the fact that I didn't really understand this until I started working for the company, but we're more related to fungi than we are plants. So that, the things that are in fungi should actually work better for us than actually herbal type medicines as well. So,
Hmm.
yeah, no, we worked on lion's mane, some of the other ones, lots of good biotrophics in there for, you know, uh, brain stuff, immune stuff. I think as time goes on, you're going to see a lot more work in this area for sure. And not just for the mushrooms that make you see things.
Right, right. I'd be very interested in knowing as well, like how much of the things when they're preserved, um, actually carry through. Because I mean, I feel like I used to make a joke about, look, for those of you who know Tim Ferriss and the 4 Hour Work Week, anybody that's watched his podcast as well, he constantly is talking about mushroom tea. And I'm just like, look, as crazy as the guy is about having ads for mushroom tea, I I don't know that it really has any benefits. I certainly don't enjoy drinking mushroom tea. That's not something
No,
that I'm
I
super
don't
into.
blame you. And I worked for a company that was one of their products that kind of made that. So, I agree. I think I think the promise in mushrooms comes with small molecules, and that what you need are things that can cross the blood brain barrier. Extracting by water is just one way. I think a lot of the really interesting bioactives in mushrooms are probably slightly hydrophilic or hydrophobic. I mean, so extracting with like ethanol or some kind of alcohol or actually CO2, I think would be a really interesting one. But yeah, that's kind of what we would do is we would do different extractions on the mushrooms, you know, and basically take those extractions, do certain experience. I'm trying not to give anything away or
Right. Good. Yeah. No.
valid,
Sure.
validate NDAs, but
Right.
basically, basically break down the mushroom chemically and then see if any of those parts actually had any validity.
Very interesting. So, I mean, I would imagine that your day is consumed, for the most part, working directly with clients on, various projects. How big is Bioinfo Solutions? How many people, work with you? And then also, what does a typical day look like?
Sure. Um, we're pretty small, as you can imagine. It's the computer is mighty, so the individual can do a lot of stuff. I would say I probably have maybe three or four that I consistently work with, but um, doing this for like 20 years, I've gotten to know a lot of people. So I think that's the thing with bioinformatics is that it's not just like another specialty. It's bioinformatics is huge. You can actually do absolutely everything. You can do, there's transcriptomics, there's proteomics, there's mass spec, there's, looking at the DNA sequence, GWAS, right? There's just so many different areas that there's no way one person can specialize in any of those. So, or all of those. So what I do is I get a lot of people that, are interested in bioinformatics. I do a lot of workshops so that I'll go and what happens is, I'll give a workshop on bioinformatics. And then at the end, I'll have students come to me and say, I really want to do this. I don't want to go in the lab and, sex mice, at 5 a.m. for an experiment. I want to just be at home, working and, actually doing research there. So I get a lot of students that actually want to help out. And so I work with them. But yeah, a typical day, usually I have a few meetings, talking to clients. I think it's very important as a bioinformatician, and I'm going to stress this, is to understand the biology,
Mm.
right? I came from the bench, I've worked in labs. I do understand the biology that whenever you do an experiment, all these genes that you're looking at actually have a history, right? They have buddies, they have enemies, within these genes. And to be able to understand that a priori and then put that information together, I think makes a better model than just coming into it and just looking at it as just regular data. A lot of my job, and I say this when I give workshops, most of my job is taking data that taking a lot of data and putting it into a format that I can actually look at. So 80% of my job is probably just massaging the data so I can actually analyze it. So some of it's kind of mindless, you can do this anywhere. A lot of times I'll get bored at home. I'll, go to a coffee shop and, work or, wherever. So well,
Pretty
it
funny.
is a lot of, part of it is, when I teach this, have you ever seen the first Karate Kid,
Yes.
right?
Yeah.
Movie, right? That kind of wax on, wax off, and he's painting the fence, and he's like, "Why am I doing this?" This is kind of, that's, when I get somebody that wants to do bioinformatics, usually I have them, annotate, massage, put together the data, and that's the waxing on, waxing off. But as you put all this information together, what you find is, you start learning more about the study, and that, you know, for me to put together data and to figure out the variables, a lot of times I'll have to go in and actually read the paper. So
Mm.
you're reading these papers, and if I say I'm working on sepsis, I end up reading like 5 or 6 different sepsis papers, and at the end, I get a better idea of what's going on, and I understand the data sets itself and what it can and can't do, if that makes sense.
Yeah, it makes a lot of sense to me. So the one thing we probably haven't talked a lot about in different podcast interviews, but so I'm a Lean Six Sigma Master Black Belt as well, and one of the things that I oftentimes would tell people, I'm like, look, the statistics tell you a lot, but the conversations around, like, the data tell me a lot more. You're like, why am I seeing certain things at different
Yes.
times? Why do I see, certain peaks and, workloads and other things, different defects happening? Hey, look, we switched a machine or whatever. I mean, all of a sudden it's like, okay, so now now I understand why we're seeing what we're seeing. But to your same point, and we oftentimes sort of take that long tail, and chop it off and go, look, I'm not even going to look there, because most of the stuff that I need to find is in the massive data that I see on the other end. And so that's what I sort of interpret from a lot of the things that you're saying.
Oh, absolutely. When you see, for me, when you see a paper come out on, say it's a big, transcriptomics or proteomics study, it's like when you see the paper, that's like the tip of the iceberg, right? And then you have all this lovely, gorgeous data underneath with, 90% like, so for me, I look into, these databases, gene expression omnibus, I look at a bunch of, icebergs that haven't been discovered, so absolutely. There is so much data out there, and that, even in a manuscript, even in a long manuscript, there's no way you can describe it all. So,
Hmm.
so yeah, there is absolutely hidden gems in just about every study that you look at. I will also say there's a lot of garbage out there as well. And you don't realize it, it's pretty easy from a transcriptomic perspective to figure out like males and females in a study. And a lot of times you'll look at a study, you'll download it, and the individuals that they say are males and females are not that. There'll be mistakes. And when that happens, you're like, how well can I trust this study? And
Right.
sometimes that happens in, studies that are, been popular, published in really, famous journals too. So
Mhm.
it's crazy.
Yeah, I mean, look, I know that mistakes can happen, but you talked earlier about sort of collections of data and the fact that there are places that you can go where there are past studies that were ran and things like that. Do you worry about that? Sort of taking other people's data and studies, and whether or not if they would actually um, be able to be replicated.
Yes, that's a huge issue. I have analyzed people's, the interesting thing, I come in, I have that post perspective, right? Like as you're in as they're in the thick of their experiment, they're just kind of, a lot of times they'll be focused on whatever they obviously wrote their grant for probably, and so we've got to
Mhm.
definitely focus in on that. But, I'll get the paper, I'll download the data, analyze it, and I'll come up with something completely different. And a lot of
Hmm.
times I'll actually reach out to them and say, "Hey, did you know this was in your data?" Sometimes they're very happy that I did that, and they're like, "Oh my gosh, I can't believe that's great," and that might be a potential collaboration. But a lot of times they're very upset that you're actually looking at their data.
Hmm.
You would be amazed at how many researchers out there get very upset with what I do. And I've presented at conferences and had like these older professors stand up and just yell at me like, "You're
Oh
stealing
wow.
our data. You're ruining everything."
Uh It's a shame that that's the case. I mean, I know at least for, I don't know, my PhD research, I was happy whenever all of a sudden people were reaching out, wanting to reuse a study that I had. I was like, hey, this is actually great to know that it lives on, and that somebody else is finding some use in the thing that I did, that I didn't think that was all that valuable in the end. So
Yeah.
that's a good thing. So.
I will say it's more of a, from my end, I get more value out of taking lots and lots of studies. So in a
Mhm.
way, they usually typically don't get mad at me because, they might have their data might be, N of one in, like 10 studies I looked at. So I would say I think that's the best way actually to look at this data, is that, kind of a wisdom of crowd kind of thing. I can't remember the guy. He did the website 538 Silver.
Don't know
Matt.
that one.
Oh, I don't know. He's a kind of a famous statistician, but in 2012 he predicted all the just about all the elections in this country in 2012. I think the only one he didn't get was like one in North Dakota because it didn't have like very much data on it. And so everybody was like, how did he do this? And, he didn't ask anybody who they were voting for. He just basically took all the surveys out there, right? Some obviously leaned left, some leaned right, and took the average of it. And it was the exact, prediction of the election. And that's kind of what I do with these studies, right? All these studies on say breast cancer. You take them all and then you combine them, and then kind of right in the middle is where your answer wants to be. So,
Yeah.
and I do reference everybody if I actually use somebody else's study in for a paper or a manuscript, I absolutely reference them. And in fact, I was very bored during COVID-19, like I'm sure a lot of people were. And as a bioinformatician, what's great about me is like, I'm going to investigate that. So what I did is just start downloading a bunch of coronavirus data and, a couple, and got some friends that I knew and, hey, you want to figure out this disease? Maybe we can save everybody.
Very
Didn't
cool.
happen.
Yeah.
But anyway, we basically wrote in this pretty amazing paper based on a related virus in mice, and the things that we were getting were actually absolutely coming out in real time with the in COVID-19 with the
Mm.
case studies and the people that actually the epidemic itself. So.
Very cool. So let's talk a little bit about your methodology and the outcomes that you typically have. What are some of the more exciting, projects that you've worked on?
I would say I think the COVID-19 thing I think was really interesting in that it just kind of showed me that you have this new epidemic. There's obviously no data on it. How do you answer questions on a disease you've never seen? And so being able to find a related virus, find a related organism where you could actually take lung tissue, which is the actual tissue that's affected, and extrapolate that to humans and have the things we found in mice work in humans. I thought was amazing. Like that was a pretty profound kind of experience that I had there. I think the honey truffle thing was really interesting. I would say the thing that I'm finding interesting is that gene expression, in my opinion, kind of it's almost like a language,
Mm.
and if you treat it like a language, you understand it better. You can think of all these different cell types as maybe different countries, right? And they maybe all have Latin as their base, but they the actual words are different, right? And different cells produce different genes, so they have different words to communicate with, but they all have kind of the underlying same basic language. So as I go through my career and I look at more and more you know transcriptomic studies, it's starting to become a language, and that when you see experiments and these certain genes pop up, you're like, oh my gosh, it's TNF, the system's going down, or oh, NF kappa B, this thing is really pissed off, this thing isn't really happy at all. But yeah, I think the more I look at the data, the more I kind of see that kind of language analogy, and that I'm very excited that I feel like I'm starting to get to the point where I can actually understand it. It's like the Matrix movies. I'm sure you've seen those.
Yep.
When Neo's in the Matrix, and they're looking at the screen, and the zeros and ones are going down. He's like, "Oh no, Nate! Neo's in trouble." That's
Right.
kind of how
Right.
I feel this gene expression stuff.
Very cool.
So yeah, I'm very excited about just not only that we're getting a lot of information, but we're also at the same time we can use these computers. If you understand the underlying biology, you can use this AI to kind of actually translate this. Imagine with a expression experiment, like Google Translate, when you're looking, scrolling down your thing and you see a foreign language, you hit Google Translate. Like that's what I'm hoping for. And eventually I think we'll kind of get there.
Really cool. If you were to sort of project out and look into the future, um, are you excited about any one, you know, new technology that's coming? I mean, I think you're leading into it with your, um, sort of what you were just saying with regards to AI. But are you excited? As things are changing in the field, or are you seeing any big changes that you think will impact us in the next few years?
I do. And this is kind of where I'm kind of taking our company is, like I said, a language isn't represented on based a table, right? And when I first started out, you do an experiment and all the genes would go on a table and they'd probably be ranked by either p-value or fold change, right? And then you just look at the table and go, oh yeah. And then you take the top one and that's usually your target. But what is really exciting me, and I've started to get into quite a bit lately, is networking. Using networks to represent gene expression. That all these genes have, like I said, buddies and enemies, and how do you represent that visually? And not only how do you represent that visually, but then how do you compare these different networks that you build? So I think for me, the exciting thing is you know I think the thing that always pushes me is how do I present the data so that when I work for a client, I can show them a picture and they get it, right?
Mm.
And I think sometimes with the networking, to see something and seeing groups here and here and how that might change with a disease, I think that's kind of the future of where we're kind of going with this stuff. So, death to tables, long live networks.
Yeah, well, I mean, I find it interesting because I think I have mentioned this on other podcast episodes as well. But so I mean, the one reason why my team gets to stay so small is because the ability for using AI automation, and all sorts of tools to be able to do things that I would need human beings for. Now I primarily sort of try and think about, do I need a physical person to engage here or not? There's certain things like in the digital marketing space, you certainly need people to do copywriting. They have to go through and kind of look at the stuff that AI generates. Otherwise, it all appears too robotic and things like that. But at the same time, I mean, to your very good point, the ability for AI to have a brain that is only centered on one part of industry, let's say, to say say to it, hey, look, you are somebody who is a specialist in bioinformatics, and you are only to post things that relate to this and nothing else. You don't know anything else, any other area. Is something that we can train AI for today. So to me, it's the same sort of thing. It's like, hey, look, the ability of these technologies to be able to do these things today is pretty impressive.
Yeah, absolutely. And what I would say is I honestly I hate programming. I hate coding. I'd rather have things shoved under my nails than worry about my syntax, whether my dang program worked. But I think that's another exciting thing is AI. I've started to meet people that they'll write a program and I'll say, "Oh man, that was a great program. Like, how'd you do it?" And he's like, "I just told the computer, I need this, this, this." And so
Yeah.
I think that's what it used to be. That was something that would bar you from bioinformatics, is not being able to program, right?
Mm.
Got to put together all these pipelines. But I think now with AI, it's giving you the ability to basically put together these programs without having to learn the computer languages, which is nice. And anybody that's studied R will appreciate this. They're like, I wish I had a program that would just let me do R without me having to talk to R. But
Oh, that's funny.
I do give this example a lot when I teach, is that you know IBM, remember their first supercomputer? I think they called it Big Blue, right?
Hmm.
And to show it off, would go, they challenged the greatest chess player, right? And then Big Blue eventually beat the best chess player. And of course everybody was like, "Oh, look, machine is better than, humans," right? But what they don't understand is if that chess player had a computer with him, as he was playing Big Blue, he would have beat Big Blue, right?
Sure.
It's not either computer or human, right? It's if you put them both together, they're better than both, right? And that's kind of how I approach this. And I think the wise thing is that if you give the computer everything and you put everything in a black box, I think eventually you'll be very disappointed, especially in biology. So having somebody that understands the biology and also the technology that you can bring to the table, I think is very important.
Yeah, I mean, I completely agree with you on the programming front as well. I'm not a programmer either, but at the same time, um, I've been able to write prompts that essentially, hey, look, I'm looking for this. Give me a JSON that
Yeah.
it will extract this information, and all of a sudden it's there, and I put it back into the thing that I'm working with, and lo and behold, I have the data that I need. So, yeah, I frequently do that same thing where I'm like, look, I don't know how to do this myself, and certainly there are enough forums and things on the internet where you can oftentimes ask simple questions whenever you run into a true problem that AI can't fix. But I find very few problems where I can't sort of keep asking questions and eventually get to the answer.
Right.
Yeah.
Yep. No, it's that's what I want AI to do, is to do everything I don't want to do. And in fact, right now I've got one of my I do a lot of internships with the students, and so I have a student right now who's basically trying to come up with an AI solution to help us, annotate data better, right? To go
Hmm.
into these databases, pull the associated information. You have no idea how, like, they'll do an experiment on humans, but then they won't give you either the sex or the age. Like, that's not important. Like, why would you not include that information? Then
Right.
sometimes if you actually read the paper, you can find that information. So, training an AI to go in there, basically look at the data and then actually go into the paper and then find the relevant information would be awesome. That would
Yeah.
save me 80% of my time. So,
Very good. So, whenever you look at the things that people need in terms of important skills and concepts, are there any particular skills or concepts that you would suggest that people really need to learn in the as they go through their education to get started in life sciences?
oh, yeah, absolutely. I think a thorough understanding of statistics is very valuable. And I constantly go back onto my statistics that, when you're doing, sometimes 50,000 different t-tests, there's a limit to that. There's you're breaking certain laws when you do that, and so you have to, realize that you're going to generate random noise. And how do I get rid of that random noise? I think not only statistics, but I think the ability to graph the data. That, in my mind, if I can actually visually see the data, that I have a better understanding of what's good and what's bad, I guess, if that
Mm,
makes sense. I don't know if you ever heard of PCA plots. They're called principal component analysis plots, and they're awesome. It's a way to take lots and lots of data and basically simplify it in three-dimensional space, so that things that are like each other are closer, and then things that are more disalike are farther away on the graph. And again, if you can visually see this gives you an idea of that, of what your experiment is doing, right? If I'm doing an experiment, I'm looking at two conditions and all my dots are in one mass in the middle, that's a bad sign. But right, if the two conditions are farther apart on the graph, then I have an idea, like, yeah, this probably worked, and I'm going to see a lot of differences. So,
yeah. And I mean, sometimes it's just so important to be able to visually interpret, the data as well, and so such a critical thing, and being able to know what tools you can and cannot use. I often used to do rough tests where I would do average of averages, which would drive people nuts. And I'm like, look, yes, I understand that from a statistical standpoint, this I'm breaking some laws, but at the end of the day, all I want to know is like, am I heading in the right direction or am I not? And
Right.
this is going to tell me whether or not if I'm going in the right direction or not. And then I can use more refined true methods once I get down to something that I actually want to look at. So
Yeah, that's true. I also kind of use a story too in that, if say you're on a football team and you have this great play design, but you throw it to your receiver, he fumbles it, somebody picks it up, he fumbles, and eventually somebody runs it in, and you score, right? It wasn't pretty, but it's still a score, right? Like as long as like with the honey truffle gene thing, right? As long as I found it, it doesn't matter how I did it, So
yeah.
yeah, it's I find a lot of people that are in bioinformatics, they spend a lot of time building their statistical hammer, right?
Mm,
And they're going to use that hammer on everything. And I, I guess my biggest piece of advice is to find lots of different tools because not everything is a nail, right?
right.
So I think that's probably benefited me the most is don't specialize, have a broad palette of things that you can use and utilize. I see so many people that just specialize, like I said, they build that hammer and bygone it, they're going to use it on everything that they see. There's a lot of interesting tools out there, a lot of interesting ideas. You know, go find those, go find different platforms. Sometimes you don't even have to come up with them yourself, right? It's um, somebody will develop something that's very useful maybe for a specific project, use it for that, but you know investigate all these tools and use the proper ones for the proper job.
Yeah. That was so important. What's the greatest piece of business or leadership advice that you've ever received?
um, I did think about that one. I would say it wasn't given to me personally. I think there was a statistician. I love when statisticians become famous because that almost never ever happens. So if they did like that's they did something important. But Georgia, I can't remember his name. I'm horrible with names too. I thought I wrote this down, but anyway, he had this famous British statistician, 1970s, said, all models are wrong, but some are useful. And for me, that was the best advice I've ever received. Is that as a bioinformatician, you're trying to recreate biology and mathematics, right? And you'll never be perfect at it. And if you can realize you'll never be perfect, but you can make it just good enough, that's okay, right? So in my mind, it's kind of like what we were talking about. You know, as long as you score a touchdown, it doesn't matter. So utilize all kinds of different things and realize that you're never ever going to replicate the biology, but you can get really close enough so that it'll be very useful.
Very good. Yeah, it looks like it was George E.P. Box. I Googled
Yes,
it
George
while you were
Fox. Yes.
there you go.
Dang
There you go. So for anybody that's looking for that one, Mike, what inspires you?
I would say working with the next generation. I believe. I don't just do bioinformatics. I also coach rugby at a high school. Like you spend a lifetime gaining all this information, and if it dies with you, it's lost, right? So what inspires me is you know seeing the next generation and inspiring them to take whatever we were doing and going farther, right? That's what I want.
We're gonna have to talk about this rugby thing because my grandson is has been going to middle school. But before that, in grade school, when they would go out on the playground, those kids were not allowed to play tag. They weren't allowed to run after each other or chase each other on the playground. I was like, this is how you learn all of these things in terms of, hierarchy and how to, how to better, sort of play with individuals and, where teaming, becomes important. I mean, is on the playground and, here you're saying that there's a place in Colorado where people play rugby. So I'm like,
Oh,
I'd love to
it's
football
pretty
about
big
that.
actually. Yes, you should. There's a lot of youth rugby. Goes to high school, men's clubs. Actually, Colorado's a pretty hot area for rugby. So,
Excellent. What concerns you?
um, like I said, I think utilizing AI in inappropriate conditions. I think that's what concerns me, and I see this a lot. Is that anybody that learns how to program or use R decides that they're a bioinformatician. And so there's a lot of, I would say, people in my field now that say they're bioinformaticians but don't understand the biology and are just kind of looking at the data ad hoc. So
Mm,
it is, I worry that, you know, how do I distinguish myself above somebody that just knows how to program, I guess,
sure.
So yeah, I would say that concerns me. And obviously with the funding going on, are we going to fund science anymore? Or if we're not going to fund it through academia, is all our science going to be funded through corporations? And if that happens, then, you don't necessarily get cures anymore. You get treatments. So
Mhm.
I guess, we got to decide, you know, is it valuable to have unbiased data out there that we can utilize? I guess what I really worry about is them shutting off the spigot for all the data that's out there. That
Yeah.
at some point they're going to say, you know what, you can publish, but you don't have to show anybody your data, even though taxpayers paid for your study. You can just keep that. So
Wow.
I guess that would scare me, is that when we stop showing other people our data. That's what scares me.
Yeah. Definitely would help the scientific advancement just in general, just being able to access, more information about things and then not being able to do that, certainly would provide a definite cliff that a lot of people would look into the abyss and go, okay, so where did all the data go? Well, maybe it dried up with somebody making a change in the way that we currently fund that research.
Yeah, absolutely. Yeah, it's I do worry about that. And that's the thing. There is so much data out there that, like I said, I kind of view bioinformatics as making a picture, and that all this data out there is kind of pixels in that big picture. And the more pixels you have, the better picture you get. And I worry about those pixels disappearing. Right now, this is kind of the glorious time. I mean, if you're in science, you should get into bioinformatics because you can research anything you want. So I would encourage all the scientists out there to pick up some bioinformatics, in that whatever you're researching, you're probably not the first person to do it. Or even if you are, there's probably something very related you could basically learn from.
Very good. Last question. What excites you?
Uh, what I fear and what excites me is probably the same thing. AI, right? I love the fact that you know I'll be able to process information. To make these networks requires an incredible amount of computing power. I guess what excites me is using the computers to visualize the data so that I can, in completely different ways. I guess would would be what excites me.
To that same point, the thing that just ran through my head as well that I think would be, really exciting, overall is I mean, what I've seen in life sciences with experiments is that, oftentimes we'll say, hey, look, we need to, you know, um, run this pipeline for X amount of hours to be able to get to the result that we want to find. And then, could we get to a faster answer potentially? if we buy more compute power, then potentially we could get to a faster answer. It will be interesting to see whether or not if AI at some point could, if you could go to AI and say, hey, look, here's essentially my pipeline, or here's the data that I want to analyze. This is how I was thinking about analyzing it. Is this the most efficient way to do it, so that I get to the fastest answer possible? I don't know necessarily that that's the right question to ask AI, because sometimes it will give you an answer that you
Yeah.
don't want. I was using this near Christmas time. I was saying, I was in Chattanooga, Tennessee, and I had asked Google if there were Michelin star restaurants in Chattanooga, Tennessee. Just a curiosity that I had in the conversation that I was having at that point in time. And the one thing that came back was that there were 3 in that area. There are no Michelin star restaurants in Chattanooga, Tennessee. The AI on Google's side was essentially hallucinating because of the way I asked the question.
Yeah. You got to be careful of what you ask. You might get the answer.
You might get an answer that you didn't want. So maybe
Yes.
the fastest answer isn't the right answer. Maybe taking your time with the data might be the right answer.
Right.
Well, Mike Edwards, thank you so much for being on the Life Science Success Podcast. I appreciate you being here and for telling us so much about bioinformatics and your view of the world. I greatly appreciate your time.
Well, thank you. It was a pleasure being here, and yeah, thanks for allowing me to be on your program.
Thanks a lot.